Okay, I'll start the session. So I've watched the progression of this aspect of medicine over the last 10 or 20 years. We started off first with cells, particularly stem cells, that didn't really work, and I'll go into it in my own lecture why. And now we're in a situation where we've got just signal messengers, and everybody's believing that they have the same effect as cells, which they haven't. Signal messengers don't really restructure tissue. They're just signal messengers, nothing more. And certainly when we disrupt tissue with, be it dermapens or whatever, we're integrating them into a repair mechanism. So as a consequence, signal messengers can have some effect, but they're probably a little bit overblown in terms of what their effect is. And that's only because the present generation of doctors aren't used to 10 years ago when we were dealing with stem cells that we got to work. So up first is, Viral, I couldn't have that because it's removed that. No, it shows the, thanks. Okay, so Amit is first, polynucleotides versus secretomes. Thanks.
Okay, good afternoon. It's a pleasure to be back to IMCAS, and this probably is the hot topic these days, polynucleotides and exosomes. First of all, the usual disclaimer. Can you cut down the light a little? Can you have the light down a little? Okay, so what I'll be doing is I'll be kind of comparing polynucleotides versus exosomes and try and tell you where are we as of now. It's kind of a debatable topic, and obviously we have to understand that these are two new kids on the block. They are, comparisons between the two is like comparing the rest of the intestine with the appendix, in the sense that polynucleotides would be the appendix as of now, with exosomes which have so many indications per se. So polynucleotides per se are, or for that matter, both of them are regenerative injectable treatments, just to give you a brief outline of it. But they differ significantly in their composition, in the mechanism, the evidence, and the regulatory status. While polynucleotides are purified DNA fragments, whereas exosomes are basically the extracellular vesicles. As Patrick mentioned, they are secreted by cells, and they're kind of a delivery or messenger particles. Polynucleotides as such have a kind of a specific role as far as the skin regeneration is concerned. So you have stimulation of fibroblasts, they improve the tissue repair, hydration, and collagen, whereas the exosomes give you the growth factors which they deliver to a particular area where you want to. But again, that's a big question mark. And I'll try and go back and forth and try and kind of give you positives and negatives as well. So as I said, polynucleotides have science behind them and have specific indications, which are skin hydration, hyperpigmentation, rejuvenation as of now. But exosomes, on the other hand, kind of have thousands of indications, and we're still kind of evaluating, right from dermatological to cardiology to maybe even neurological indications, but none very specific as of now. So this is the latest update that you have. If you again compare the two, PN versus exosomes, PN has good clinical evidence for skin rejuvenation. Whatever literature is available, it's approved and widely used in most of the countries, and generally excellent safety profile. And if you compare it to exosomes, they are promising, but the evidence is still got to come. It has to be standardized. There's a regulatory landscape which is very unclear, and we are worried about this last point, the source processing and the quality control. So you have the human base, the animal base, the plant base, and we're still not sure where is it coming from. So as I said, I'll play the devil's advocate and try and go back and forth with the positives and the negatives of both of these products. Remember, when you communicate, you got to dispel the delusions and illusions that people have about a particular product. So I start with a handicap here, that the utility of PN per se in skin rejuvenation has been kind of better kind of defined, but it does not have the wide indications that exosomes have. So you got to overcome your nervousness when you're using newer particles or newer molecules and try and kind of get into a confidence building. So let's kind of understand in a simple term, what are these? As in, polynucleotides for me is like Amazon Prime. You go on there, buy a product, and it gets delivered to you. whereas your exosomes are more like your grab food or porter floating around. You can kind of ask them to deliver everything and anything, and not really sure whether it would come in the right way or the right manner. So you ought to understand that we are not disguising things. We ought not to disguise products, be very specific, and that's where the polynucleotide probably scores over the exosomes again. And we have to be safe. Remember, we are doctors, we can't be injecting patients. So polynucleotides are something that we inject. Whatever is injectable is always has a better safety profile, a better efficacy. Whereas exosomes, would you still like to inject? There's still question marks there. We're still applying it topically post microneedling and hoping that someday we might get to inject them as well. The biggest worry for me and in practice is the risks that are there with exosomes. And in fact, most of the patients do come in and ask about, when they read about it on the net, they're worried about the infection risk, especially when it comes from the source of the infection. The second point is the most important one, the unwanted long-term effects. That is what worries most of the patients. And look at the line at the end when it says promotes the growth of precancerous cells. So you have to be careful. Don't get into an argument, whether it's your workplace or your home for that matter, which you see on the left side. Don't get into an argument regarding these products. It has to be scientifically based. You've got to get your product in the right manner to the right people, and the right literature has to back it. Now let's, as I said, I'll be the devil's advocate. So here you have another article which comes in, which talks about how they're actually not prone to immunogenicity. Exosomes, according to this particular article, is produced naturally within the human body, so they would not be prone to immunogenicity, so therefore they should not elicit a host response. So we have kind of counter views which are floating around. Coming to delivery techniques, when you're doing it in the clinic, the delivery techniques are important. With PN, we are kind of sure we're injecting it intradermal, sometimes subdermal, but exosomes are something that can wander around. And if you're lucky, they might deliver whatever you're sending to the cancer cells, and then it's a kind of a gala dinner, which would happen tomorrow night for them probably. It is something that you should know. In practice, as I said, you should know where to inject, how to inject. You can't probably end up doing what has happened here on the right side. The zoo actually ended up discovering that the hyenas they were trying to mate were actually two males. So be clear about your indications as well. So again, going back to the counter view, the recent studies have actually now shown that the therapeutic viability of these loaded exosomes can be target specific. So there's one view which says they can wander around. The other view says now that it can be target delivery. So in a way, it's getting better with time for us as well. And the other thing is the longevity of the result, the amount injected, the sessions that are required. Polynucleotides, you just inject once or twice, and you can expect results as far as the skin rejuvenation is concerned up to 6 months. Whereas exosomes require a lot of injections, a lot of sessions which can trouble the patient. So again, specificity is very important, which is more for the polynucleotides. So polynucleotides, you get more predictable outcomes, usually 4 to 6 months after a course. Exosomes, you have greater variability. The variability is long-term data limited, so we are not sure when is the effect coming. We're all kind of getting into protocols that we are devising in clinic and trying to get to the final answer. So as of now, polynucleotides is driven by science and not being pushed down your throat. But if you go around in the trade area tomorrow or today, you'll see exosomes as a big money-churning machine, and it's money being pumped in to make us believe that this is probably the best. So sometimes whatever you see is not right, and whatever your peers tell you may be the right thing. Maybe the wife doesn't know everything after all. So final tips and takeaways. Polynucleotides, as I said, consistent results, definite delivery style. The transitions are seamless and more professional protocols are coming in. Exosomes, you need to seek your feedbacks. We ask our fellow friends, doctors, what is the response, reflect on the performance, then we're exploring new techniques, setting personal goals, and we are iterating and adapting according to the need. And again, counter view, regardless of their clinical indication, exosomes, if they follow GMP practices, I believe they would probably work. And we've seen in practice, especially for the hair rejuvenation or the hair restoration, exosomes is one thing that is working quite well in practice, not as well as in the skin rejuvenation. But the world already knows some facts. So these are two kind of messages from on the left side. A patient of mine, when I suggested exosomes to her, she said there's no FDA-approved exosome products on the market. In fact, this is just an FDA-authorized clinical trial that they're doing as of now. And then the right one that you see is a doctors group where they are talking about how USA has banned the Korean products and talking about them as just being fragmented junk proteins and scattered growth factors. So still there's a dispute regarding exosome which exists. Finally, clinical application. This is what is the kind of final outcome. Polynucleotides do work in periorbital rejuvenation, fine lines, acne scars, neck rejuvenation, post-laser healing, and the laxity. Exosomes, we found hair restoration is probably the key thing where they really work. Not too great as far as the skin rejuvenation is concerned and still experimental. Thank you very much, and I invite you to COSDOM India and Mumbai. These are the international faculties joining us. Do join us in Mumbai. Thank you very much.
Thank you so much, Amit. And of course, you opened a Pandora's box where everybody will have different opinions out here too. But next we have Dr. Diala, and she's going to be speaking beyond SPF towards personalized photoprotection driven by technology, skin biology, and of course, exosomes.
Yeah, good morning, everyone. So let's discuss the future of personalization in cosmetics and aesthetic medicine now. So we are shifting from a one-size-fits-all approach to a true precision dermatology powered by AI-driven skin analysis. This enables a transition from reactive care to predictive and preventative strategies, allowing earlier and more personalized interventions. Ultimately, we are no longer treating skin types, we are decoding our skin biology. If we have two patients, same treatment, completely different outcomes, this is the limitation of our standardized common protocols. Personalization is essential here because skin is highly variable between individuals, from UV response to aging patterns and inflammatory behaviors. These differences are driven by complex interaction between genetics, epigenetics, the microbiome, and the exposome. Understanding this variability allows us to move toward more precise, effective, and truly personalized treatments. Cosmetogenomics allows us to understand how genetic predispositions influence key skin functions such as pigmentation, DNA repair, and oxidative stress response. At this time, epigenetics also explains how external factors like UV exposure can accelerate biological aging. Importantly, these changes are not fixed. Many are reversible, opening the door to targeted and personalized interventions. Cosmetoproteomics, it's all about how we can reveal our ongoing damage. This allows for highly precise, dynamic treatment strategies tailored to our skin current biological state. We can't miss the opportunity to talk about AI here. AI brings together imaging, molecular data, and environmental exposure to create a comprehensive view of the skin. By integrating these layers, it enables more accurate prediction of skin evolution and treatment outcomes. Ultimately, this leads to a deeper personalization and a continuous optimization of these aesthetic and dermatological procedures. The intelligent mirror brings also here a real-time skin analysis into our daily practice, assessing texture, pigmentation, and hydration instantly. By integrating AI with exposome data, it provides a deeper, more contextual understanding of skin behavior. This enables dynamic, continuously personalized skincare recommendation tailored to each individual. Photoprotection becomes here a personalized biological intervention. Personalized photoprotection moves beyond SPF alone toward a biology-driven approach tailored to our individual skin needs. It integrates multiple layers from topical protection with antioxidants to systemic support and microbiome modulation. This strategy also adapts to environmental exposure, enabling more precise and effective skin protection. We move forward from treating aging to managing longevity. The Skin Longevity Index here is an integrative model designed to capture the full complexity of skin aging and health. It combines genomics, epigenetics, proteomics, microbiome, and exposome data into a unified framework. This approach enables a more precise, predictive, and personalized strategy for long-term skin longevity. Additionally, wearable technologies capable of continuously monitoring parameters such as UV exposure, hydration, and oxidative stress to enable a truly personalized and preventive skin care. Combined with innovations like skin organoids and predictive AI-driven models, these advances may transform dermatology from a reactive specialty into a proactive and adaptive science of skin longevity. We are transforming and transitioning our practice from a static understanding of skin to a dynamic, continuously evolving model. From genetic approaches, we move toward a true personalized strategy based on our individual biologies. And from corrective treatments, we advance to predictive and preventive care, shaping the future of dermatology. Thank you.
Thank you so much. Thank you for the wonderful talk.
Sure. Our next speaker is Patrick, and sorry, can I just have this? Patrick, I know you were talking about the uses of exosomes within dermatology and recent advances. Thank you.
Good afternoon, everyone. I'm Patrick Huang, a dermatologist from Kaohsiung, Taiwan. It's a really pleasure to be here and share this session with my fellow panelists and who are all world-class experts in regenerative medicine. So I'm deeply honored that the World Congress of Dermatology has appointed Dr. Achima from Thailand and me as two of the Asian Pacific ambassadors. Together, we'll represent and promote the voice of the dermatology community across the Asian Pacific region. I would like to invite you to speak for Asian at World Congress of Dermatology in Mexico next year. So today, I will be sharing update of on the rapid evolving topic exosome in dermatology, recent advances, misconceptions, and clinical insights. Here's my conflict of interest disclosure. Although I work with several companies in the regenerative medicine field, today's lectures is not sponsored by any of them. So with that, let's move on to today's topic. This consensus statement published in February 2024 was developed by more than 1,000 experts worldwide. Importantly, the author recommended using the term small extracellular vehicles or small EVs for vesicles smaller than 200 nanometers rather than broadly label them as exosome. So once again, this reminds us that the terminology matters, especially in a rapidly evolving field such as regenerative medicine. So as of December 2024, a total of 16 clinical trials involving exosome therapy for skin disease and regenerative medicine were registered on clinicaltrial.gov. And now there are 25 registered clinical trials worldwide. What's interesting is shift in research focus. While earlier studies centered on wound healing and regeneration, and now seeing more and more trials about hair restoration. This is quite similar to the previous presentation about the trend. So in fact, the number of hair-focused trial has been increased from 7 to 11 just since this year. So I would like to recommend this April special issue of the Essay Surgery Journal, not because our papers are included, but because it reflects where the field is today, and with some of the latest evidence, debates, and an uncertain question surrounding regenerative aesthetics. So I would like to list 10 misconceptions about exosome in dermatology. So some people ask, why isn't my exosome product showing in the research comparison? So does that mean it's less used or less effective? And actually, this is a common misunderstanding. In this recent publication, actually, researchers analyzed 12 clinical trials using exosome derived from ADSC or platelet, but none of them from commercial products. That's because of most of commercial exosomes actually are protected by their proprietary manufacturing and the confidential data, so making them difficult to include in academic comparison. So the second misconception is some people ask, are university-derived exosomes always more reliable? I once asked a researcher from top university in Taiwan the same silly question. This is the second misconception, and she loved me and said that he actually wish that he could work in a big company where they have the most advanced equipment and the control system to produce the purest exosome. So academic doesn't mean always better. So in fact, industrial lab often ensure higher consistency and quality. So you often see advertisement claiming their products containing billions or even trillions of exosomes. So some people ask, particle count matter more than purity? This is the third misconception. Purification standards for exosome vary worldwide. So actually, some products reach 99% purity, while others consider 70% acceptable. So many people think that have more exosome particle automatically means a better result. But actually, higher count actually doesn't means more purity, because they also contain a lot of impurity, like the lipoprotein, protein aggregates, or cell debris. So that's why big number doesn't mean more effective. Some people think that the production capability doesn't affect the exosome quality, that quality and quantity are unrelated, but this is not completely true. So large-scale production actually means a strict process control and batch consistency, and also advanced purification system, which can actually improve quality and reproducibility. And on the other hand, small-scale or a lab-made batch may be pure, but they often less stable and harder to standardize for clinical use. So many people also believe that using a patient's own exosome is always safer and more effective, but this is not necessarily true. So many patients see using their own blood or autologous EVs, which is the new product available in the EU, as the most natural and preferred option. The earlier science actually was very promising, but we need more large-scale clinical trial to prove effectiveness, and also need standard consistent way to collect and measure these EVs. So especially according to this study, only about 0.5% of what we isolate are true exosomes. The rest, about 99% are lipoprotein or protein aggregates. So which means that it's very hard for us sometimes to identify which particle is not exosome. So furthermore, autologous exosomes also are found less potency than stem cell exosome. And very interesting that most people, many people actually thought the PRP exosome is safer. However, once the exosomes are processed and re-injected, they are still have some safety concern. So aging does not only affect the cell themselves, it also affects the EV. They release. So when cell age, the microRNA, proteins, the lipid carry within EV also change significantly. And some of the vesicle actually may also contain some senescence-associated signal. So if this is true for many other cell types, it also raises some question that should we expect platelet-derived exosomes from older individuals to have the same regenerative potential as those from younger cells? So some people believe that if the exosome work well, then injecting them intravenously must give even better result, but this is not true. Not about the safety, but about this, because that we know that most of injected exosomes, they are relatively clear by the liver, spleen, and the lung within minutes. So very few actually reach the target tissue. So that's why intravenous injection is not, does not bring some effect, but also higher risk, because we know that exosome probably can carry some immunosuppressive effect. So that's the reason why also increase the risk of the infection and also the cancer risk. And some people worry that exosome can cause cancer in reality. Actually, exosome does not create cancer cell, but they are natural messenger released by almost every single healthy cell. But actually, that exosome from healthy stem cells can, while some of the studies shows that it exert anti-cancer effect via some so-called ceramide and the microRNA-mediated apoptosis. But tumor-derived exosome can have the opposite role. They support cancer progression, so that their impact actually very higher, highly source dependent. So, but this one doesn't mean we can inject exosome for cancer treatment. Yeah, because we still know nothing about the effect of the exosome if we deliver systemically. So I think one thing should be clear by now is exosomes are not stem cells. They are biologically active on their own, and their effect depend heavily on where they come from. And different cells produce different exosomes and carrying different signal and ultimately leading to different clinical outcomes. So in this study, the author directly compared umbilical cord mesenchymal stem cell and the adipose tissue mesenchymal stem cell exosome across multiple biologic function. And this conclusion was straightforward. Both have regenerative potential, but they are not interchangeable. AD-MSC exosomes are more effective at promoting angiogenesis and the tissue remodeling, while umbilical cord MSC exosome appear to be stronger anti-inflammatory agent. So this reinforces an important concept. Not all exosomes are the same, and their biologic effect depends largely on the cell they come from. No matter how natural they are, regulation is still essential, because there are many things we still don't fully understand. So that's why I strongly support strict government oversight to ensure their safe and responsible use. So interestingly, as exosomes become increasingly popular, regulators around the world are start to respond. So countries like Indonesia, Thailand, China, they have introduced stricter regulation last year. And in Taiwan, many non-human exosome products are now classified as EV. This is very interesting because the people thought that exosome is more well-known, so they renamed the non-human derived exosome become EV. That's how the Taiwanese government do. So, well, the role of exosome in dermatology, including wound healing, hair regrowth, and aging, and hyperpigmentation. But because of time limit, I will just show the publication we have since last year. So this is something about pediatric morphea treated with laser-assisted delivery of the exosome. Then you can see that the hair, after 2 application, till now, there's no further hair loss, and the regrowth actually surprised all of us. And this is a publication about gray hair turning black following exosome treatment. And in our, so this is our publication. If you are interested, you can take a look. And overall, the result is quite encouraging, and 60% of the patients achieving more than 50% improvement. So this is the one actually published in April this year, just mentioned the special issue on ASJ. So this is one thing very interesting. In this patient actually with special gland dominant skin, both sides improved initially. However, by 6 months after 2 treatment sessions, the side treated with microneedle IV alone showing noticeable better result, which is a different result than what we expect. The orange line represents microneedle IV alone, and the blue line represents treatment combined with exosome. Interestingly, the orange line continues to improve throughout the 6 months follow-up, ultimately showing greater pore reduction than the exosome treated side. So this was not what we initially expected. It suggests that in patients with special gland dominant skin, exosome may not always enhance pore tightening outcome, and in some cases may even attenuate them. So last but not least, exosomes are not a replacement for what we already have. So they are innovative option, especially when conventional treatment fails short. So let's take a close look at how exosome might help us solving a problem what we once thought are unsolvable. So this is the conclusion. Exosome clearly hold tremendous promise for regenerative medicine. If we can achieve the future of this field will depend on scientific rigor, standardization, and the patient safety. So if we can achieve that, exosome may become one of the most important regeneration tools in modern dermatology. Thank you so much for your attention.
Thank you for the wonderful talk, Patrick. And now we again have Patrick now talking about secretosomes, the new kid on the block.
Thanks for having me, and Patrick, thanks for a wonderful lecture. We have to, I think, I've been given the envious task of deciding whether exosomes or secretomes are the most important. I think the first thing we need to do is just look at the differences in the way medicine's going. We've got aesthetic medicine, we've got regenerative medicine, we've got longevity, and they're 3 different things, and they're all beginning to become confused. These are some books that I'm having coming down the line that are going to address the different things, particularly with peptides. So we got to understand first that exosomes and secretomes are nothing more than signaling messengers. And this is so important. They're not regenerative medicine, and they only have a role in it if we damage the skin and use them as a repair mechanism. If we consider for a moment hospital colleagues, they deal with degenerative medicine. They wait till things go wrong and then they try and fix them. We deal with regenerative medicine. And I've been doing this, I suppose, for 20 years or more. If a child falls and cuts their knee and you put stitches in, the body heals itself. That's regenerative medicine. If you fall and break your leg and an orthopedic surgeon puts in a plate, he isn't doing anything in terms of repair except bringing things together. That's body regenerative medicine. So back in 2003, I decided to use what orthopedic surgeons and dentists were doing and take PRP and use it for hair. And we used first PRP, myself and Viral here set up clinic in India 2007, and that was the first FUE in India. And we turned around then and used red light with it, years before people were writing papers about it. So if you consider American politics for a moment, you've got the Democrats and Republicans fighting with each other. And what are they fighting over? 3 things. Infrastructure, whether to waste money on wars or whether to repair the roads, particularly some of the roads like Miami Airport is falling apart. Anyway, that's another day's work. the body does the same thing. I'm only mentioning it because signal messengers are the people that direct the parties. Where the DT or sort of Hillary turns around and says something, they're the signal messengers. Also, if you look at the American Army at the moment, that's the same thing as immunology. So you've got infrastructure, cell repair, you've got how you use your defense forces, and the third thing, of course, is communication between cells. Communication, do you teach critical race theory in your third level education or do you not? So left and right will have different attitudes, and this is what signal messengers are, how cells communicate with each other. But the thing is, that has nothing to do with how the cell reacts. If you take a city like Cape Town, or many other cities, I lived in that, you've got to take into account corruption. That's chronic inflammation. This is what we should be dealing with. And people talking about putting IV NAD into the body, that sets up inflammatory reactions that do more harm than good to patients. So the big thing that all signal messengers work at is repair, communication, and defense. They all do that, be it exosomes, PRP, growth hormones, cytokines, they're just signal messengers, no more. So what are secretomes exactly? So secretomes in many ways are a combination of everything. They include secretomes, exosomes, and of course all the other things. So growth hormones are nothing more than just the 7 growth hormones. And don't forget as well, we're priding ourselves in what they can do with skin, hair, and vaginal repair. These are easy. If I slap somebody in the head, you can almost make hair grow. If you derma roll somebody's head, which is what we used to do, hair will grow. If you use RF in a vagina, suddenly female stress incontinence disappears. They're easy to do. So is skin. Why is skin so easy to do? Because dermal rets fall into epidermal rets. We used to do it with lasers. So if you damage one, it'll go across and go from the dermis up. It's much different doing décolletage, where they're even together. We ran the first ones in 2007, how to do that. So what are exosomes? Exosomes, you know yourself at this stage, they're sort of extracellular vesicles, and they go to a specific point. So in other words, they're almost like a postman bringing a signal to a specific address. They're almost like those bombs flying from Iran into Tel Aviv city. They're going GPS guided to something. If a bomb goes off in a market square in Baghdad, that is like a secretome. It has no respect for anybody or anything. It's just a general thing. It's not taking out one person. And they're the totality of all messengers. And that's why I sort of mentioned them like that. So don't forget, they also contain exosomes. So if you consider, it's almost like going into a restaurant, and if you order a meal of hamburgers and chips, that's a secretome. If you order a hamburger on its own, that's an exosome. And I only mention this because we're probably better off using secretomes in here rather than exosomes, because secretomes can set up the skin in a way that exosomes can't. They can reduce inflammation. They can do all those things. That exosomes are only using specific things. So could I have some sound on that, please? So if, well, we're not going to get it. So I'm looking at, we say, as a human orchestra, and if the secretome, have you got sound? Sorry, our guys can't understand English. But anyway, I'll explain to you. If you take an orchestra, the conductor's baton is what the direction of what's going to happen is, what part of the orchestra. If you have got a situation where exosomes are almost like a violin just playing that rich tapestry per se, and it's too late now anyway, I told them before. Yeah. So, are secretomes something new? No. We've been using secretomes. I did molecular biology a long time ago, and we were well aware of secretomes then. They just have become familiar in the recent thing because obviously exosomes and secretomes are quite profitable for different companies. I'll give you a little bit of history. We tried stem cells originally in New York, Cornell University. They tried them for myocardial infarctions. This sort of worked, not very well. Then in Tel Aviv, we used them to repair spinal cord, and they didn't really work at all. They just made big blobs of tissue. And why? Because they were cells. The body reacted against them. And as a consequence, when the body reacted against them, you didn't get differentiation of tissue. So we looked at this back in 2010, 2017, and we took stem cells originally from stromal factions because we were doing a lot of liposuction, but later from a commercial brand. And we got them to work fine because we used red light with them, PBM. And that set up originally what I thought was the right environment, but it was doing a different thing completely. If you take a Krebs cycle and spin it for the biochemist minutes, it forms 2 units of energy. If you turn around and use red light, believe it or not, it makes 36 units of energy. And how does it do this? What it does is it allows the cytochrome C to release nitric oxide and oxygen binds. Oxygen binds 20 times the amount that it does in a normal cell. And what happens to nitric oxide? It's a gas, so it escapes from the cell. A very potent vasodilator for those among us in the audience that use sildenafil or Cialis. So as a consequence of that, it is perfect for wound repair. You see in some of my other lectures how we reversed it within 10 days grade 3 burns that the hospitals were sending us, and many other things. And the hospitals really run quite behind in all this. So you've got a situation where if you use red light with stem cells, and I don't see why they don't do it at exosomes, there seems to be such a normal progression to do, you've got a situation where not only will a wound, and why does a wound not heal itself? 2 reasons. Particularly you see it in burns and vascular occlusions, because there's no blood. And if there's no blood, there's no repair mechanisms. So it's left to God to try and treat it, and he forms a big dermal scar to protect the tissue underneath. But common sense would say, if you stand back and say, okay, I'll take over and do this, and you give a situation where you give stem cells with red light, you will get epidermal repair for many reasons. One of the biggest reasons is the release of nitric oxide from the cells itself. And you can measure this. You can go online and get a swab you put in your mouth in a little thing, and you can see when you use photobiomodulation that you actually get the release of the nitric oxide. It's very measurable, and I don't know why nobody's doing it. And you can also add in methylene blue, and it even works better. But you have to be synchronous with your time with methylene blue, because if you do one after the other, you crowd the stem cell. You have to understand how mitochondria work. Mitochondria, when they start flagging, what happens is they release electrons into electron transport chain, and they get blocked. That's the problem. So if you try, even though they don't work intravenously, but if you even try to use enzymatic methods of blockage so you get more, let's say, NAD within the cell, it doesn't also work because the cell is just blocked. So we can use methylene blue as a shunt mechanism to flee up the electron transport chain, and then everything flows down the line. Anyway, that's another day's work. So secretomes per se, we went through that. There are similarities between them. Obviously, they're both signal messengers. They're both biological. They both can modulate cell behavior, as I said earlier, within immune, within communication, within defense. And they have obvious differences as well. One of the biggest things is mode of release. Exosomes are derived from an endocytic pathway mechanism, whereas secretomes originate within the secretory pathway cell. So secretomes often leave the cell and as a consequence interact on other cells. Exosomes obviously need a signal messenger receptor base to enter a cell, so it's a bit more difficult for them to do that. As I say, there's nothing new about secretomes. We've been using them a long, long time, but as people moved away from using stem cells, unfortunately, we will get it right and we will get stem cells to work. The problem is that we overrated them in what they could do instead of preparing the territory they were going into. And one of the big things as well, obviously, about using red light is it reduces inflammation and reduces the ability of other cells to actually knock them out. So in the short term, secretomes, because they're not cell-based, have a slight advantage in doing things that stem cells didn't do per se. So how am I doing time-wise for all? I have a couple of minutes left, okay. So at the moment, they're obviously gaining a lot of attention, but unfortunately, people who are selling them are hijacking this aspect of medicine to make us believe that they're going to be involved in longevity. They're not, trust me. My new book coming out on pathways of aging, which looks at epigenetic drift, methylation of DNA, which looks at mitochondrial dysfunction, which looks at cell senescence, all of them, we're still missing one big point. Of the 7 forms of the pathways of aging, we're assuming that if we put in one thing, it'll improve it. It doesn't. They're all integrated together. So as a consequence, if you influence one, you affect the other. So we're going to be 20 years at this, and it'll be the next generation of doctors, trust me, that will bring this all forward. So it's wonderful to have all the enthusiasm out there, but we cannot let influencers on social media influence our young doctors to convince them just because they say something that is going to work. Most of these people don't have an education in science like we do, and we should leave it to the scientists to tell us. Now, we have one big problem, obviously, with peptides, because peptides are natural within the body. They can't be patented, and if they can't be patented, then the drug companies can make no money out of them. So it's their advantage to say that these things don't work. And Robert Kennedy, if there's one thing he did, which is probably the only thing he did, which was to benefit science, and that is to bring it back to compounding pharmacists to do this, because it has been hijacked by a lot of Chinese companies that are just sending us things. Peptides will work, and they'll work better than exosomes or secretomes, because they're directed to do a function, whereas exosomes are not. Exosomes are directed to just tell it what it should do. Whether it does it or not is a totally different thing. And then the other thing, of course, is exosomes' ability is to change their cargo. So what we need to do is stop praising ourselves that we make hair grow or that sort of we make vaginas not leak anymore, but to look at people who are in nursing homes that are 1/3 of the population of this room will end up with neurodegenerative disease. We should be working on inflammation and how to reduce it. And there's no doubt about it, if we change the cargo within exosomes, we will treat pancreatic cancer, emphysema, all those things. At the moment, we're just taking what's in our own body and giving it back to us. So we're just amplifying the signals that were in our body already. That's a totally different thing than putting proper microRNA and all within the cargo of an exosome and make it do things that's going to make people live longer. What am I at? Okay, so as I say, at the moment...
Patrick, I think we need...
okay, I'll wrap up there. So what they will do at the moment is what we see on the screen there, but I advise you, if you really want to get these things to work, consider red light. And it's not only red light per se, it can enter the brain through your skull and you can reduce inflammation per se. Okay, I'll wrap it up at that, and thank you very much indeed.
Thank you so much for that lovely talk. And next we have Dr. Diana, who's going to talk about exosomes used for post-care topical application after lasers and EBD.
So we have this for short people, right? Is there a way to make these slides bigger so that I can see what's on them? Because they're so small, I can't see. Oh, much better. Thank you so much. Good afternoon, and thank you all for coming. My topic today is, can post-care regeneratives change outcomes? And I think we all know that exosomes and a lot of the secretome products that Patrick has been talking about can reduce the duration of post-energy-based device treatments, but the change in outcomes or improvement in quality of outcomes has been yet unproven. So I'm a plastic surgeon. I've been around for a while, like Jane and Patrick know. And I started plastic surgery in 1990. And at the time, we were still doing things like phenol peels. And during the phenol peel and the early erbium and CO2 lasers time, we just covered up the wound and hoped that it would heal underneath. We did complete wound care coverage, put on occlusive dressings. We might or might not get an infection. And of course, this took forever to heal. And the patients just knew that they had to hide out for a good couple of weeks. Once we evolved into the laser era, then this was more of a structured thing. It wasn't adhesive tape, but mostly gauze. We knew to keep the area wet because there was some science that showed that moisture helped reepithelialization quite a bit. But even so, the postoperative care was very elongated, and sometimes people developed problems such as hypertrophic scarring, areas of third-degree skin loss, and even fungal infections. Once we entered the fractional era, not all the skin was damaged, and we started having patients who didn't need those occlusive dressings. Recovery times were quite a bit quicker, but still pretty significant downtime, and I think on average, even when we entered the fractional era, let's say this is 10 or 12 years ago, it was still a minimum of 5 days. 5 days seemed to be the magic number, and nobody really wants to hide out for a weekend, much less 5 days. So if we look at the segment of the wound healing cascade, which goes from the hemostatic phase to the inflammatory phase, and now we're in the proliferative phase, which is several days afterwards. This is when we see fibroblast expansion and ECM deposition, which are influenced by many things, including decorin, fibronectin, the fibrillar proteins, et cetera. We're also looking at neoangiogenesis and reepithelialization. So this is a critical phase for recovery. How can we accelerate this from days to weeks to hours? This is where our topicals come in. So there's a really good study by H.H. Kwan, and this is one of the few numeric recovery day phase, as well as the outcome enhancement studies that showed in a laser patient population and a split face construction, laser plus exosomes applied topically, decreased the ECCA score, which is a quantification, weight-based quantification of severity of acne scars. The ASCE versus control showed 32.5% improvement over 19.9. So this is one of the very few split-face studies that supports enhanced outcomes. If we look at my friend and colleague Melinda Kimbrell's study, which was published in, Patrick mentioned the special edition on exosomes, which I was lucky enough to be one of the editors of. So Melinda looked at a series of over 100 patients that she did plasma resurfacing for and found that she decreased the reepithelialization time for these pretty severe and aggressive cases down to 9 days, from 13 to 14. And her outcomes improved greatly. You can see that the complication rate without topical exosomes versus with exosomes decreased from 13%, pretty high adverse event rate, down to 3%. All of these articles are in the ASJ Special Edition on Exosomes, published 2 months ago, and this is all open access. So if you want to read the whole thing, I think it's a great resource. If we look at exosomes and reepithelialization, and there is a systematic review, then we can see that the exosomal influence on keratinocyte migration, of course, essential in post-care, following things like microneedling, laser treatments, et cetera, and as well fibroblast function really improved with the use of topical exosomes. This is my own patient, and don't share her picture, but you can definitely take the post-care plan home. And I've developed this over the period of about a year and a half because I'm doing quite a few laser treatments, anything from a very superficial 10-micron procedure to multiple layers and a combination of fractional and ablation. So this patient had one of the most aggressive treatments that I do, and she was 100% re-epithelialized, not without pattern marks, but 100% new skin coverage at 14 hours on day 1 post-treatment. And so this is a pretty rigid pattern of treatment. I have the patient do 5 saline soaks followed by topical application of exosome serum, and as well an occlusive emollient-based exobalm, which I compound myself. And so basically the series is 20 minutes of a cotton cloth saline soak, followed by the serum, followed by the emollient balm, about 2 hours apart. The next day, the patient will wake up with not so much swelling. You can see that her eyes are swollen, but not swollen shut. And she wouldn't really go out and engage in any social events, but at 24 hours, she was able to go to the grocery store with basically a minor sunburn type of look. And at 48 hours, she could go out with topical tinted sunscreen, which I think is probably more rapid than anything that I've experienced with aggressive treatments. What about the use of ablative laser? This is a patient that I treated sebaceous hyperplasia and her upper lip with 80% ablation. And I did 8 layers or 800 microns on her nose and 500 on her upper lip to get rid of or significantly improve sebaceous hyperplasia and vertical lip lines. It was interesting in using this program that these areas healed at the same rate as fractional laser. And I've not seen that before. To me, the quality of outcome is better. I did not treat her paranasal and nasolabial fold area with ablation because I didn't know about the healing rate, and if I had to do it all over again, I would definitely treat the pores and the thicker areas of skin, like on the chin and the nasolabial fold region, with ablation. If we look at this JCD article and look at the efficacy of combined treatment with human adipose-derived stem cell exosome-containing solution, this is a 12-week prospective randomized study, then we can see that the outcome, 28% of the patients were rated over a GAIS-4 with the exosomes as compared to 14% with the saline control side. Here is a QR code if you guys want to scan the QR code for the Exosome Journal, then we would be most happy for you to read all our articles. We had 12 peer-reviewed articles, and I think this is probably one of the biggest projects that we have done community-wide for the study of exosomes in regenerative medicine. So this is a patient of mine. Let me see if I can you start my video, please? On this patient, I can't. Maybe I can do it. So this is a patient of mine that had precancerous lesions on his skin and really severe acne scarring. I treated him with 4 layers of laser coring, dermal treatment and superficial full ablation. And here he is at 2 weeks post-treatment, and you can see pretty significant skin tightening. So this, to me, is a new arena of the combination of regeneratives and fractional laser. So I think there are some limitations, though, of exosome therapy posed to energy-based devices. As Patrick pointed out, the exosomes do not make new matrix. You need a matrixome for that. You can get shortened recovery, and you can get enhanced outcomes. But because the primary mechanism of action is reduction in inflammation, scar management, and differentiation of the path of healing from the N1 inflammatory to the M2 anti-inflammatory pathway, again, the exosomes are signaling agents. They are not substance. They don't build tissue. So I think that's the biggest limitation of exosomes, but I do use them on every energy-based device treatment, and many of the ones that are that do require a recovery time, they are best combined with other modalities of treatment and not a single agent. Thank you so much.
Thanks for that, Dan. Some wonderful results. By the way, you're using sublingual arnica. Is that for bruising or for edema, or?
It's mostly for edema, but it's also for the erythema, because erythema is one of the inflammatory agents.
Oh, yeah, it is anti-inflammatory. I must. Thanks for the tip. I must try that myself.
It's cheap.
And across the counter, yeah. So, Jane's up next, Jane Yu, and she's going to tell us, I suppose, to bring some biological honesty back to exosomes in the 2026 period. Thanks, Jane.
Thank you so much for being here to the very end. My name is Jane Yu, and it's always humbling to go after Diane Duncan. So I hope to enlighten you today about exosomes and bring to you some things that you haven't heard, but also some things to reiterate. Okay, so 2026, the hype cycle is over. Now what next? I have no disclosures. So extracellular vesicle biology, this is the foundation we need. What they carry, of course, mRNA, miRNA, as well as other growth factors and other lipids and DNA fragments. I think that we really need to understand the science and the biology of these vesicle transporters and really look at the fundamental mechanisms in which they affect the processes that we're looking to achieve. Now, Patrick mentioned a little bit about this, the MISEV 2023, which is a consensus statement. The International Society for Extracellular Vesicles discourages exosomes. It recommended that it be relabeled as small extracellular vesicles. Why? And they wanted to understand how we can come to some sort of understanding about the particle size and count, different surface markers, the purity, the potency assay, because all of these things are not necessarily standardized currently in the literature. What does the market sell? Well, the products that are currently labeled as exosomes include adipose MSC-derived extracellular vesicles, as well as platelets, umbilical cord, amniotic fluid, as well as Warden jelly. But all of these are not equivalent, so we really need to be able to have true clinical comparisons. So where are we? I think, from 2020 to 2023, we had a really, really great hype cycle and everything was overinflated. Now we're kind of at the trough of disillusionment. We're from 2024 to 2026, where we kind of like lowered our expectations, but now we're kind of plateauing and going on this slope of enlightenment, trying to figure out, well, where do exosomes really play in our procedure base? Okay, well, FDA enforcement is accelerating. It's not slowing down anytime soon. Previously, I spoke about different warning letters, especially the Chimera that came out in 2023. In late 2024, it was CHAR-EXO as well as EXO-RNA. Early 2026, the DOJ has some criminal referrals. Okay, so this is pretty serious. And in March, the FDA has updated with some consumer warning letters. So this is something important for you guys to notice as administrating physicians, because you share in this liability, okay? So sourcing is a patient safety and medical legal risk here. What do we know about some of the issues here? First, again, as the nomenclature, as I alluded to, there's batch-to-batch inconsistency, undefined potency assays, and heterogeneous sourcing. So sometimes, exosomes is really more of a marketing term and not a product specification. Secondly, the regulatory cliff. There are 0 FDA-approved exosome products, and there are many warning letters that have come to existence. And currently, what are the evidence gaps? Well, there's only 21 studies in the most comprehensive 2026 systematic review. These are small, uncontrolled, and heterogeneous. And we have very limited skin of color data. Most of the studies have not been performed on darker skin populations, and no long-term safety data. Well, what is the clinical evidence? I think they're looking through all the PubMed sources from 2024 to 2026. We have things that are definitely promising. Other speakers mentioned hair restoration, pigmentation, as well as acne scars and atrophic scars. One thing that I found is really interesting is for facial rejuvenation, that we had a split face study looking at PRP as well as exosomes, and exosomes did perform very well. So there are really great instances where exosomes can be highlighted and do what it actually are meant to do. But the jury is still out on some things like photoaging as well as hyperpigmentation/melasma. And of course, just a reminder that there is no legal approval of injected exosomes in the United States. Not all exosomes are created equal, as you know. There's human, plant-derived, as well as platelet-derived exosomes, and they all vary. We spoke a little bit about autologous exosomes, and some people think it's the holy grail. Why? Because you're drawing from your own, the patient's blood, fat, and serum, isolating these extra vesicles and then reapplying to themselves. Autologous material is immune compatible and it's personalized to the patient, so this is something that's totally safe. The closest analogies that we have include PRP. However, there are some technical barriers that exist with this. One is the yield. So the blood only yields a certain number of extracellular vesicles, and the therapeutic dose is unclear. And so you have the adipose that yields higher levels, but then requires lipoaspiration. The characterization, there's no way really to characterize it at the time of getting them. And then the standardization, because every EV cargo differs by a person's age, their gender, their disease state, as well as medication. So you can't reproduce it from session to session. And then there's a regulatory gap here. So the clinical status in 2026 is that true autologous exosome vesicle therapy remains investigational. PRP is the most practical proxy that we have. Secretomes. This is something that is not new. This is something that has been around for years and years, but I think this is something that we all think of as like, okay, this is the newest thing since exosomes. No, we've known this since the time of stem cells. The secretome is a complete bioactive output of the cell. So what does it include? Exosomes, microvesicles, soluble proteins, growth factors, cytokines, matrix components, metabolites. It's a whole kitchen sink and more. So this, in some sense, may be more therapeutically complete than any pure EV fraction alone. And there's more investigation being done in this arena with umbilical cord as well as adipose-derived stem cells. And there's some debate right now about using whole conditioned media versus purified EVs. Part 2, what does the future hold? Well, there's engineered and designer EVs. I'm really excited about this because we can now genetically modify and load and have things that are specifically targeted and have different things within the cargo elements. So one of the things that is really exciting is having something that has an exosome that is a carrier with botulinum toxin. So this can overexpress the SV2C botulinum A receptor, and it comes preloaded with botulinum toxin. So imagine that, and it goes specifically targeted. We also have different AI-designed EV platforms where they optimize the cargo loading based on a person's own genetic information. And then finally, the hydrogel EV delivery systems, because drug delivery is a barrier, right? So this ensures that the EVs reach the target dermis as opposed to being degraded. Diane spoke about EVs and energy-based devices. This is a real area that is coming up, and the EBDs create this controlled wound environment. So EVs have a real opportunity to play a role with RF microneedling, fractional CO2 laser, the standalone microneedling, and sonophoresis as well as ionophoresis. I just want to reiterate that for photoaging, the mechanisms matter. So you want to look at the pathways, whether it's downregulating MMP expression, increasing collagen elastin, regulating antioxidant stress. You want to know that whatever cargo is affecting the specific pathway involved, tyrosine pathway for melanogenesis, for instance. So this is just a framework for you as an evidence-based clinician to use. I think we have a lot of data for things like topical hair restoration, but you want to verify the FDA registration and GMP compliance, and there's certain things that, again, don't go into those fuzzy areas with some companies that have gotten warning letters from the FDA. So these are just some of the things to tell your patients. I saw exosome injections on Instagram. Are they safe? Well, there's real science, but the challenge is that there's zero FDA approved for injection. So the topical procedures right now are very safe, but be cautious about anyone offering injections outside a clinical trial. Now, are they better than PRP? We kind of discussed that there was a split phase study. Exosomes may have a consistency advantage, but the PRP has more longer safety data. Can I use exosomes I bought online? Well, the over-the-counter exosomes are largely unregulated, so I would say follow the evidence. And then what about exosomes for my melasma? Well, this is an area that we're still watching. Now, what does the field need next? I think there's different priorities. First priority includes more randomized controlled trials. Second, skin of color representation. There needs to be more standardized delivery protocols and long-term safety follow-up. And then looking towards the future, autologous and patient-specific platforms should be available. And with AI, we should have assisted formulation as well as targeting. Now what? Well, I urge you to have a 3-point commitment. Vet before you adopt, so require the characterization of anything that you get in your office. 2, speak precisely. Investigation is not FDA cleared or proven, so this is what you should be talking to when you talk about these products to your patients. And then finally, I encourage you all to be involved in clinical trials, case reports, case studies. Generate the data that we're missing, because it's only with your help that we can bring all the evidence together and push the field forward. Thank you.
Thanks for that, Jen. So we're into the Q&A session, and I'll run the first question by you. What evidence is there to support the fact that when we're microneedling exosomes into skin, that we're seeing a true signaling effect rather than just wound repair itself? Because nobody has even set up the experiments against each other. No. There's no evidence at all, is there?
There's no evidence.
It's shocking, isn't it? That here we are all buying exosomes and even the basic experiments aren't being done. Are there any questions from the audience? Ron, I'm sure you have something interesting for us.
I'm stunned. I'm observing all this very carefully. But no, I don't have any insightful questions.
Does everybody in the audience use exosomes, or somebody not? For those that are using exosomes, how many find that injectable are much better at working than rolling them into the skin? Because I started off injecting them and I thought it was much better, to be honest, particularly with hair. Amir as well, yeah. I found that as well, that the original human ones are better, even though I know the reason the plant ones will work, and I found injectable were better than rolling them in. So believe it or not, I've resorted back now, getting my staff, because we do I once had about 20 hair transplant clinics, and as I got out of that, with their suggestion, went into non-surgical, and we tried them all, and we're back to the way we were 10 years ago with stem cells and red light. Viral, have you any questions for the audience?
I think the question you can answer, will autologous exosomes have equal effect as much as the ones which are obtained either from amniotic or placental or other sources?
Yeah, I noticed in India you're using a lot of the heat-defined ones for autologous. I used different types of autologous, and to be honest, I didn't find them much better than what we were doing. One of the problems I found was exosomes are 50 to 150 micrometers, but I mean, the autologous ones are using 600 and above. So I mean, that has to be their filter mechanism, really secretomes rather than exosomes, but that's just my own personal opinion. Diane, have you anything to contribute?
A couple of things. I think that in looking at the autologous exosomes or the autologous secretome, I know there are a couple of new devices out there in the US. We have a device, ExoCube, or the PRP kits that extract the autologous platelet-derived secretome exosomes. Most of the, if you look at the early phase of injury response, it's the hemostatic phase, and that's when the platelet-derived products, mostly growth factors, are expressed. But those, when they're used in a prolonged manner, can give you more of an inflammatory response or pro-inflammatory. So you might want to use the platelets early on, or the PRP products early on, but you don't want to use those as a sustained thing because they're pro-inflammatory. Another thing that, there's a US product that uses stored platelets. There's a thing called platelet storage lesion that really degrades the quality of platelet-derived exosomes. Another factor, and I don't know, I know you probably have an input on this, is that the age of the donor does really matter. That I find
totally.
The umbilical cord products are much more
and
effective.
what medicines they're on as well.
And so if you have a person with hair loss and they're 65 years old, you're probably not going to get much benefit
Sure,
from
sure.
autologous exosomes.
I found as well that we were told that we could charge 3 times more for autologous, and we only have to do them every 3 months. But we resorted to changing the protocol and doing them every month like we were doing before. But there's no doubt about that. If somebody, for instance, is on steroids the day before or the day off, yeah, you're wasting your time, you know. And even a lot of cardiac drugs can suppress the response as well. Amir, what's your... Sorry, Amit?
2 points. First of all, what is your protocol? This is a question. What's your protocol for the hair? Because there are different protocols floating around. How many sessions do you do? You said every month. So how many sessions are you doing?
Okay, so what we normally have been doing was originally we used PRP and stem cells. We're using them every 3 or 4 weeks for 3 months and then putting them on a 6-month maintenance, and that worked fine. But we were told that when we used autologous, we could drop that back to every 3 months, and we found at the end of 6 months, it was almost like the growth that you would see with normal ones after about 2 months, you know, so we reversed our protocol back. I think we were oversold, you know, because I know the company we got it from have changed their protocol back closer to ours now, so.
Do you think the protocol ought to be a kind of a faster protocol, as in do it every 15 days?
To be honest, Amit, I think they're using the wrong technique completely. I think you have to use ultracentrifuge spin cycles of probably up to 20,000 to 100,000 rather than 2,500 in order to get exosomes off properly. I think all we're dealing with is nothing more than probably PRP and things mixed with it, you know, because all evidence in a scientific lab is if you want to get exosomes, you've got to spin them up in the hundreds of thousands, you know.
And how do you compare the plant-based, the bacterial, and the human? Obviously, human is the best as of now, what I understand. But what is your opinion on the 3?
I think if we delude ourselves that just because things are plants that they don't work very well, they've been around 40 million years before we were are, and as a consequence, it does work well. It's easier for the companies to make them because they're very stable compared to exosomes per se. One of the problems, I suppose, with exosomes per se is that a lot of the companies are actually forming the lipoic structure around it. And the University of Dallas in Texas showed that a high percentage of what we're told as exosomes that have went through that lipidization phase, 24% don't work at all. And that's sad, you know. Let alone what we're being told as exosomes may not really be exosomes. There's no proper standardization,
Right.
you know.
Would you say that umbilical cord derived, because they are the youngest stem cells, exosomes would probably work better than the other sources?
Interesting question. The stem cells we were using were those ones from Singapore, the commercial ones from red deer, mesenchymal style cells, and I found they work fine. But to be honest, we were using them 2012, and then the company started using them for hair because we did 2016. But really, they're probably a mixture of exosomes and secretomes. Whether they're really stem cells, look, I'm a scientist as well as a doctor. They're sending it to me in packets in the post. If there were stem cells, they'd be dead because they need cold chain. So as a consequence, I think we're probably being oversold them as stem cells as well. I don't think they're real stem cells.
Hi. Any experience of using exosomes with hair transplantation in the recovery of donor area or the recipient area itself?
Give us that again. With the experience of using stem cell or exosomes for hair transplant, the donor area
And the recipient site also, both.
It's a good question.
Can I answer? Actually, we're doing a study in India, but not for the recipient, for the donor area. We're trying to do a split scalp study to see if the healing is better. It should be probably out in a year or so, but early results definitely show healing better. As Dr. Diana's paper also has said, the healing is definitely better on the side where you apply exosomes.
So let me understand. Okay, so anybody would say with androgenetic alopecia, you're going to be injecting them in obvious areas that aren't within the donor site, where you would transplant hair into normally. There's no point in injecting it into a site that has 80 to 120 follicles per square centimeter. So what is the question exactly?
No, that was what he said for both the area, recipient and the donor. So I think for donor, he means for the healing part, right?
So we have been using PRP for both the sides on regular basis, and it shows consistent improvement and a better graft uptake.
Yeah.
So can exosome be used at the recipient
How do you
side?
compare that? Unless you don't do a split scalp study, how do you compare that it's growing better? Because if you're doing 5 patients and 5 patients, the results anyways differ. This has always been my contention.
Yeah.
Well, to be honest, I did it much different when I was doing hair transplant. I dipped the follicles into the PRP before we put them back in.
Yeah, I do
and
the
I
same.
think that was very sensible.
But I still go back to what I'm saying. How do you compare, unless you don't do...
Splits.
Yes. So we are actually doing that. We are applying on one side for 5 days and leaving it. We are following it up till 14 days. Parameters are redness, erythema, healing, itching, all these, So we'll soon be out, but definitely it's showing faster healing.
So have you tried
Why should it have better wound healing? All the difference is obviously just the effect of the diodeoxyepiandrosterone within the cell itself. So, I mean, it's an effect on stem cells, which shouldn't really affect wound healing, I wouldn't have thought.
Yeah. Wound healing at the donor side should be okay. I think it should help it. But any experience with the recipient side, because it might enhance the graft uptake.
Again, the same thing. The problem is you won't get ethical committee approval for doing a split scalp study for such a thing, right? Ultimately, donor is going to catch up, so that's okay. But you won't get that study, and unless you don't do a split scalp, how do you know? Because if you do it in 10 and 10 patients, anyways the uptake is different. Anyways the healing, anyways the graft uptake is different.
Transplant? Have you used exosomes?
We do use them, okay?
okay.
I mean, I'm a believer that exosomes do work well. I've been using for nearly 6 years now.
Yeah. They're
But
more
they-
non-surgical, aren't they, really?
Yeah, they do work well. There are a lot of patients who didn't respond to PRP, did better with exosomes. So I don't have a doubt there. Yes, off record, because we do inject them, because it does give a better result. And it avoids wastage. If you microneedle, you're going to get some wastage for sure. Everything is not going to get absorbed, and not at the depth you want it to go. So yes, we do inject, preferably, but off records, because not allowed. But you get better results. In my experience, I've used synthetic, biomimetic, plant-based, everything. Human, I mean, definitely amniotic is giving better results for some reason.
Thank you.
Have you used red light with exosomes at
Pardon?
all? Have you used red light?
No, not
Nobody has. And even for biostimulatory calcium hydroxyapatite, I mean, it's a good adjunct.
I think,
Safe.
Patrick, what is your experience? The source of exosomes? It's always a big controversy.
The source of the exosome? Well, actually, about the regulation, because in the first few years, actually only plant-derived exosome was available in Taiwan. And then finally, the human-derived exosome became available last year. So I tried to see the split lesion comparison, the half plant, half human. While for the anti-inflammation effect, especially for the first few weeks, both are the same. But for the
split head?
split,
Yeah,
so for example,
hair
the patient
growth.
was very long,
Oh, yeah.
wouldn't actually just apply half for the human and the other half for the plant. I think the first few weeks are almost the same. But I'm just wondering for the second month or third month if the human will last longer. But it's still lack of long-term follow-up. So I have not confirmed yet.
Okay.
It's a tough
I've
one as well
actually-
with hair, because some of them have trifollicles, some of them are bifollicles. It's a hard one, yeah.
No, but I've actually even injected them for pigmentation. I have some results here, right here on my phone. Amazing results. Yeah, but more so with the amniotic derived.
So what is the take-home message of this session? Like, should we use it, should we not use it, or there is
whatever your country allows. Be legal, whatever you do.
No,
But-
regarding exosome in toto, should we use it, like, or should we not use it, or we still need to wait for the evidence?
I didn't hear the question.
He's saying, should we use or should we not use or wait for evidence? Well,
So exosome, so what is the message from this session today? Like, should we use exosome, or should we not use it, or should we wait for the evidence to come up?
I think we're doctors. So as a consequence, we have enough intelligence to separate nonsense from reality or hype. So exosomes are very safe. I think there's absolutely no problem with using them, but I think it's what you promise your patient is the delivery. And unfortunately, we've got one big problem, and that is because you can roll them in, it means beauticians and estheticians can do them instead of doctors or even nurses. So as a consequence, they're going to be in it for profit. That's not any way demeaning towards them. So they're going to spoil it for us because they're going to overhype it. And in Britain and Ireland particularly, it's almost moving from the medical clinics into the high street beauty salons. They're doing it. So I think we've lost that war already.
It definitely has, and I've published on complications in the past. So if anyone does have patients that they have issues with, please don't hesitate to get in contact, because myself and Marina Landau try to find you some dermatologists or plastic surgeons who can help you all over the world.
let's take a consensus. How many of us, with a show of hands, would use exosomes in practice? Okay, it's I think a little majority. And how many would inject? Very few. Okay, I think off record, how many would inject? Yeah. So I think I think there's an answer to your question. So majority feel we should use them. But how do you want to use them is a...
Sorry.
No, I'm saying, are we being recorded? I inject.
I think we all inject them. Sometimes it works, but we don't know how. I injected it for like 5 or 6 patients that I know. I told them that I was off-label, but I saw an improvement, but I don't know why or how. So I don't use it for all patients. For hair, it's good. For neck
And you can
rejuvenation.
never be sure.
Yeah.
My staff, we had a meeting, and we've about 20 staff, to decide whether to inject or not. And I said, okay, we're not allowed to by law. And then one of my senior nurses said, look, there's a guy coming for the last 2 or 3 years, let's inject him. But he had a reaction for the first time ever after the injection that wouldn't have happened the other way. And it took us quite a while to sort it out. But just that's the way the cookie crumbles, I suppose. Okay.
Okay, I think we already way over time. So, yeah.
Yeah, very nice insight from this entire session. I have an interesting observation while doing hair transplant and using exosomes in post-op, especially in the gray hairs. I have noticed reversal of graying.
There are many papers. There are some papers.
It can affect downregulation of the gene, and that has been proven, yeah.
But there are at least, I think, 4 or 5 papers which have documented this.
Yeah.
But you still can't say that it is a treatment for graying of hair. We still need much more evidence. Yeah.
True, yeah. Thank you.
Okay, thank you.
That's the end of the session. Thank you.